Well, thank you very much, everyone. I'm going to try and hold the microphone like this. Yes, I used to be a scientist once upon a time. I was a research associate, but at the moment I'm back at medical school at McGill in Montreal. So I'm afraid for the last year, the only research that I've been doing is can you turn poutine into a superfood simply by removing the gravy? Unfortunately, it's all negative data so far, but it's delicious work. So back in 2012, I published a hypothesis of obesity that suggested that the upper gut microbiota is able to detect dietary refinement, and that resulting inflammatory changes from that was behind obesity via leptin resistance. I gave it a very long title because I wanted to try and explain the whole thing within the title so that nobody could miscite me as, in some way invoking glycemic index. But ironically, that's what's happened extremely commonly. So I wanted to start off by quickly explaining why I'm not convinced by the conventional insulin carbohydrate hypothesis of obesity. For weight loss, we know that with Atkins-style diets, you need to sharply restrict carbohydrate. We also know that glycemic index is highly variable between different methodologies, between individuals, as you can see the data from this cell paper down at the bottom here, and within meal context. So both of which would suggest that if insulin theory was the main driver of overweight, that only the lowest glycemic index foods and the smallest amounts of carbohydrate could be safely eaten if you expected within a population to have no problems with overweight or metabolic issues. Of course, we know from ancestral diets that that's not what we see. The benefits appear to be macronutrient independent. There's a gradient of different consumption of meat and starches between the pole and the equator, and yet we see no parallel gradients in health outcomes being reported. And one of the peoples who are best studied have got some of the highest carbohydrate intake. 70% of the Catawban's energy intakes in the form of carbohydrate. And if you are going to try and do creative accountancy with glycemic load and portion size, that's some serious creative accountancy if you manage to hide 70% of your energy intake, not hitting the bloodstream in a way that insulin theory would predict for some people in that population would be problematic. So furthermore, in some Western populations, this data was published this year, in a study of 12,000 Finns, obesity was found to be inversely related to carbohydrate intake. Both glycemic load and sucrose intake were inversely associated. If you can see a big study like this with such apparently weird findings, if you're expecting carbohydrate to be causal, you've got one heck of a confounding factor present. And I suspect that that confounding factor is likely to be dietary refinement, which is consistent with their observation that the inverse sucrose-obesity relationship appeared stronger in high fruit consumers compared to low fruit consumers. In other words, if you're eating your carbohydrate in real food form, it doesn't seem to be associated with overweight. Dietary refinement being a bugbear is increasingly a point of agreement even among people who would gladly nuke the paleo diet from orbit. So it's increasingly perhaps one of the main disagreements between the mainstream view and the ancestral diet view that is the status of bread. I would argue that bread is our oldest processed food and may well be the anchor that holds the Mediterranean diet back every time it goes head-to-head with the Paleolithic diet in trials. So to summarize low-carbing versus macronutrient independent ancestral diets, I view that flour, sugar, and refined carbohydrates are somehow different to fruit and root vegetables in a manner that doesn't seem to be glycemic index dependent. Whereas conventional low-carbohydrate diets and ancestral-style diets both produce similar reductions in refined carbohydrates, and I'm suggesting that may be what's causing both to have their effects. At this point, I do have to point out the obligatory disclaimer that if a diabetic, you've got problems with your glucose homeostasis, and obviously it's going to benefit you if you restrict all carbohydrates. So I'm not trying to rewrite the playbook on diabetes here. So for a very quick overview, and simplified hugely, of the gastrointestinal microbiota, and I've got to point out from this point on, GI is probably referring to gastrointestinal, just in case anyone gets confused. We know that germ-free mice are resistant to most models of diet-induced obesity. In fact, the one model that they do seem to still be susceptible to has such a high level of palmitate that it's probable that it's able to directly activate TLR receptors. So it's sort of short-circuiting the same inflammatory mechanisms. We know that the microbiota is changed in obese animals and humans, and that overweight phenotype is transferable by stool transplant. We also know that you can transplant the Treg immune cells from lean to obese animals, and that has a protective effect even if the obese animal is eating an obesogenic diet. We also know that in obesity the Treg levels drop. So that together implies that there's a microbial potential causal role in obesity, and that there's also an immune system function in that chain of effect. The makeup of the GI microbiota varies with what part of the gut you're looking at. In the upper gut, and of course extremely in the stomach, there's very small numbers, but it's worth noting that even small numbers are still able to cause disease, H. pylori being a case in point. The upper GI composition is known to differ from the colonic, and it's worth pointing out that flour and sugar, of course, are absorbed before the colon, and the vagus nerve, which is thought to play a big role in energy homeostasis and the onset of obesity, projects only to the upper gut. So I'd argue that if the microbiota is going to be causal in obesity, we really need to be looking at what's going on in the upper gut, not down in the colon, where there's a broad body of evidence showing that there's inflammatory functional changes in vagal afferents and the hypothalamic nuclei with obesogenic diets. This is sort of largely through interfering with leptin signaling by a suppressor of cytokine signaling 3 and a couple of other options that have more recently been discovered. The earliest that this has been rolled back is that even in mice, after 1 day of an obesogenic diet, they're showing inflammatory changes in the vagal afferents and in the hypothalamus. Subdiaphragmatic vagotomy was able to prevent this. The upper GI microbiota is, of course, a fully feasible trigger to cause this vagal inflammation. And another clue that there's some sort of neural signaling going on in obesity and metabolic issues is that when you have a gastric bypass operation, there's such rapidity of the effects that kick in improved glucose homeostasis. It's within a matter of hours, certainly long before any appreciable weight loss has occurred. I'd argue against a clinically relevant role for obesity for colon and the fiber, because if it were down to altered energy harvest by increased efficiency by extracting fiber and turning it into short-chain fatty acids, that's surely just calories in, calories out by another route. You would expect over time homeostatic corrections to prevent an animal putting on weight because it got more effective at extracting energy from its diet. Unless, of course, there were also changes to energy homeostasis itself. Again, if the short-chain fatty acid signaling that's widely reported was really clinically significant, you'd expect dietary fiber interventions to do a lot more than just make you poop better. Again, microbiota researchers do an awful lot of their research down in the colon, and this is largely because that's where the overwhelming majority of the bacteria are, but it's also that it's much easier for them to get feces from patients than it is to stick tubes down into the duodenum. It appears I've missed out the poor old jejunum there. So it was when I was sitting next to a bucket of mice and reading the saccharine disease that the penny really dropped. Cleve was talking about the apparent absence of diverticulitis and appendicitis in ancestral populations. And he made this point, that raw foods are resistant to bacterial decomposition in as much as the cells constituting such foods are or recently were alive. There is no need to discuss here the biological mechanisms involved in this resistance. What matters is that such resistance is not present in inert foods like refined sugar. And of course, I'd been desperately pondering what could be making the difference between flour and sugar and the real foods that I'd been eating that I was doing pretty well on. And this really got me thinking, and because I was aware of the impact of the microbiota through my work in my day job, And of course, a year after I published my paper, we found out that the very bacterial changes that Cleve was proposing have actually been happening in the mouth as we've introduced new foods. In an excellent study by Alan Cooper's group from the University of Adelaide, they sequenced the oral microbiota from human skeletons dating back, I think, to over 30,000 years ago, by using the dental tartar as a time capsule of what the microbiota had been in the mouth. They were just scraping this stuff off the teeth of skeletons. And what they found was that the composition of oral bacteria changed markedly with the introduction of farming, and again around 150 years ago. And he says that with the introduction of processed sugar and flour in the Industrial Revolution, we can see a dramatically decreased diversity in our oral bacteria, allowing domination by caries causing strains. The modern mouth basically exists in a permanent disease state. So what do these real foods bring to the table that a big pile, that a Twinkie doesn't? The old foods were cellular and alive. So they're the product of the evolutionary thrust and counterthrust of animal and plant cells versus bacteria over hundreds of millions of years, each trying to get the upper hand. The evolved complexity that they've got is both structural and potentially molecular, and it's all with a view to not being an easy meal for bacteria. You can argue that if they're in cells, they're going to have a slightly delayed release, so you're only going to release the cellular contents down in the small intestine and not in the mouth. And of course, one of the best delineations between the new foods with their high carbohydrate density and the old foods, which are low carbohydrate density. By carbohydrate density, I mean the weight of carbohydrate excluding the fiber per 100 grams of weight. And that's a sort of approximation as to what kind of concentration of growth medium you might get in the small intestine at the point that they're released from the cells. And when you stack them up hierarchically, all the ancestral foods migrate to the bottom, and all the not so good foods like poutine and stuff migrate up to the top. And of course, we know the effects that these denser carbohydrates have on the oral microbiota. So you can argue the toss about whether it's going to be the cellularity, the concentration or the density of the carbohydrates, or the molecular complexity. But at the end of the day, a good point to fall back on when you're trying to think about what's good to eat or not is likely to be, does the food retain the characteristics that evolved to prevent it being degraded by microbes? In real foods, you've overwhelmingly got the possibility that yes, and in modern refined foods, absolutely not. They've been made with mouthfeel, with flavor, with how long their shelf life is, and all those things in mind, but not with any regard at all to how they're going to interact with a microbial ecosystem, let alone one that's right next to your immune system and your vagus nerve. So while I know there's a lot of argument that we've got a problem with an insufficient diversity of types of fiber and prebiotics for the microbiota, I think the overriding problem that we've induced with a Western diet is likely to be a dense nutrient broth overfeeding of the wrong type of microbes that has allowed a smaller number of species to expand its population, to proliferate at the expense of a more complex ecosystem. So it's analogous in a way to the effects you get when you have lots of fertilizer runoff into rivers and the algal blooms that mess up the rest of the ecosystem. And of course, all of this, it can actually in people be detected by nose. I've known people who were eating a pretty much paleo diet, and then one day you notice there's this sort of acrid smell on their breath, and you say, well, you've been eating bread. And they'd say, sorry. So you can actually smell this, and at the other end as well, when you're walking around the emergency room, some of the odors that from bedpans, you go, oh God. And of course, it's the same with cats. So when you've got a refined food, of course, it's also possible that the altered texture and particle size is going to further exacerbate things. This was a fascinating study that was done by Desmarchelier a couple of years ago. We've got 3 different groups of mice eating control, or high-fat, high-sugar, or high-fat chow. And when they're in pellet form, they put on differential levels of weight gain while they are eating in an ad libitum manner. But as soon as those foods are powdered, the same pellets are powdered, all 3 groups of mice were found to put on weight at exactly the same rate, and it was the same rate that they put on with the high-fat chow. And that's a real head scratcher for how that works. They tried doing different flavors of powder to see if it was palatability driven, but they didn't see many differences with that. But it was interesting that these 3 groups of mice ate the same level of calories in each case. So the mice that were eating the high-fat chow knew to eat just a little bit less by weight because it was more nutrient dense. So that would argue that there's some sort of energy homeostasis involved. So one of the options that I keep coming back to when I'm thinking about how this could possibly work is, could you have a difference to the inflammatory effect of a microbiota if it spends more of its time surrounded by large chunks of relatively inaccessible food before it finally gets broken down and digested, versus from the mouth onwards, the whole thing being at easy suspension, high concentration, bite-sized for a bacterium. So, of course, you can't try and write a unified theory of obesity unless you grasp all the nettles. So what about these very, very low-fat diets that were being tested by Kempner and others in the 70s, where there was extremely low fat, but they were still including large amounts of refined sugar? I mean, isn't that going to completely mess up the microbiota? Well, you've got to remember that what the immune system sees as the microbiota is in large part delivered by chylomicrons, with their hitching a lift across the gut barrier on chylomicrons. And if you increase your levels of dietary fat, we know that the levels of lipopolysaccharide and other microbial products that get into the bloodstream go up. So what's possibly going on here is that if you think of the barrier cells, the epithelium, with the mucus layer, the gut-associated lymphoid tissue, and of course the immune system ready to phone off to the brain via the vagus if it doesn't like what it sees. If you reduce the level of dietary fat enough to the very low levels that were being used in these test diets, it's possible, to my mind, that you could stem the flow of chylomicrons and the lipopolysaccharide and other microbial products that were moving across there, so that the immune system would see less of what was going on. In this case, what happens in the lumen would stay in the lumen, at least as far as the immune system was concerned. A recent interesting suggestion that Chassaing and Gewertz have made was of the concept of microbiota regression, which was based on some work they saw in TLR5 knockout mice don't have the flagellin receptor. They're not able to recognize certain classes of bacteria. And they also found with certain dietary emulsifiers that they got a more aggressive microbiota where more bacteria penetrated further through the mucus layer, and there were more bacteria actually in contact with the epithelial layer, where they would be detected by toll-like receptors and pattern recognition receptors. And of course, the mucus layer is tremendously important, which is being highlighted by Akkermansia is a bit of a hero, one of the ABCs, as we know, which plays a role in preserving the mucus layer function. Also, there's the brush border alkaline phosphatase. This is an enzyme on the brush border that breaks down microbial products like LPS and stops them getting inside. In mice where this was selectively knocked out, these mice developed weight and metabolic issues, which could be countered by supplementing their diet with alkaline phosphatase in the food. Of course, some other players that are likely to come up in the research soon are things like M cells that are constantly monitoring what's going on in the lumen, Paneth cells, which are manufacturing antimicrobial molecules such as defensins. I wouldn't be surprised if we don't start seeing some effects of these in various ways on obesity models. So the emulsifiers and microbiota regression idea that came from that work is very interesting, because carboxymethylcellulose and polysorbate 80 are in an awful lot of processed foods, and there were relatively and realistically low concentrations of these were found to produce obesity and Met syndrome and colitis in mice. The microbiota encroached on through the mucus and got more onto the epithelial layers, and the species present were altered. All of this was transferable by microbiota transplant to germ-free mice, which again indicates that the microbiota itself was causal in this. If you've got a processed food with some of these emulsifiers in it and lots of flour and sugar, it really is a perfect storm. People, we shouldn't be eating this stuff. I'm guilty of understatement. So this is what I think might be going on inside a hunter-gatherer. We've got living cellular starchy tubers, the microbiota in the mouth, the microbiota in the gut, and what the immune system sees are all what they've been seeing for, before we were even human. And as a result, you have a balanced immune system response, probably to other infections as well, and perhaps better immune system function in general. Accurate adaptive energy homeostasis, and a healthy weight and metabolism. As soon as a Westerner starts eating, I suspect that we know that as soon as we start eating a Western diet, the oral microbiota gets messed up. In the susceptible, this could then lead to periodontal disease. I suspect that the ileal, the upper gastrointestinal tract microbiota, will also be profoundly altered just by exposure to Western diet. What then determines whether or not you go on to develop obesity is likely to be inter-individual differences in your immune system response to that, or perhaps some factor of gut leakiness to whether it sees it. If there's a difference in European evolutionary history, it may be here or downstream from here, from our 10,000 years of exposure to flour. Of course, when a Westerner does finally start developing inflammatory changes, we see the pro-anti-inflammatory imbalance develops, the Treg numbers are known to drop. We see vagal and hypothalamic changes, inflammatory changes that cause leptin resistance and delayed signaling of satiety, and other changes, including altered energy expenditure, I believe. These would lead to obesity, which in turn generates an enormous secondary and much more widespread phase of inflammation, which would then feed forward into all manner of other conditions, including, of course, cardiovascular disease should be another big arrow over here. And again, the field of the microbiota and its role in the genesis of autoimmunity is something that's generating a lot of interest at the moment. So when one eats an ancestral diet, one would hope that the microbiota would move back at least enough towards how it would have been, a less inflammatory state, and that this would then stop the stimulatory action on the immune system, which would cause remission of everything downstream from there. I just wanted to point out, it was just published the other day, a mouse model, this one, not Welsh rugby players. Exercise, again, increasing diversity of the microbiota. So. One idea that I've had, this is a bit speculative, but it's possible that some of the proposed benefits of intermittent fasting may be simply by draining the swamp, and this may just help kickstart the transfer of the microbiota back to where you ideally would like to have it. Similarly, prebiotics, probiotics, stool transplants even may have a role in helping break locked-in ecosystem dysfunction that's persisting after years of Western diet. I suspect that, of course, you've got to have lasting dietary changes for these other interventions to work, because who has the healthiest microbiota in the world? Hunter-gatherers. Did it protect them when the Western food arrived? Absolutely not. So unfortunately, it seems to respond quite quickly to diet. So diet is key. There's a lot of questions that one asks about obesity in general. It's worth thinking about again with the microbial immune paradigm in mind. How efficacious are we expecting a real foods diet to be over time? How strictly do you have to adhere to it? Do we have bacterial adaptation? Are the bugs making biofilms to sort of hang on through the periods of good eating? Do you have immune sensitization in the host? We know that those lucky people who are always been lean are relatively resistant to the refined foods in the diet, whereas those of us who progress along the red arrow, we're pretty much left with that kind of relationship then. No matter how long we eat well, we get hit harder when we eat the bad stuff. What's the memory element of this system? Is there a lasting ecosystem change, as I say, with things hiding out in biofilms? Is it an altered innate immunity with altered patterns of toll-like receptor and pattern recognition receptors? Is there adaptive immunity involved? Do we have lasting changes in metabolic signaling? What does insulin do when inflammation's around versus when it isn't? And how does the liver process fructose when inflammation is around versus when it isn't? These are things I'd love to see looked into. And again, of course, the possibility that there are lasting neural changes due to inflammation. So last year, the first comparison was made of duodenal microbiota between lean and obese patients, 5 patients in each group. Of course, just as everyone eating a Western diet's got a pretty much messed up oral microbiota, I suspect that most of the changes have already occurred, and that in this study all the patients would have been eating a Western diet. So it's kind of hard to interpret what they find. It may even be a sort of feedback from obesity onto the microbiota. But what they did find was a higher abundance of genes that are encoding the lipid metabolism functions within the microbes, and a decrease of the ones that are encoding for carbohydrate. So it's, with all that in mind, it's a bit tough to interpret the data. What we'd really like to see done for the way forwards is to do a similar profile of the upper GI microbiota, but in patients who've been eating an ancestral diet. I mean, ideally hunter-gatherers, if you could persuade some, because you have to put a tube all the way down into the duodenum, versus humans who've been eating a Western diet. I'd expect to see analogous changes to what we see going on in the mouth, with an increased either aggression or increased immunogenicity. There's different flavors of LPS, apparently, of Western upper GI microbiota. Unfortunately, scientists in a rarefied funding environment wheel like birds evading a predator. And if you want to ask for funding to look at something that's a bit new or unconventional, you run the risk that the hawks of defunding will come and pounce on you. And due to the way science works, if you have a period of non-productivity that goes on for a few years, that may be the last grant you ever get. So it's really high stakes for scientists. And I've probably got this curve all wrong, but I blame PowerPoint because it's about the only curve it can draw. The point I'd really like to get across to people in sort of conventional dietary research is the red area is where all your guidelines come from studies that are looking at diets that still retain processed foods, refined foods in the form of bread and other baked goods. We really need to get across to them just how non-linear that curve is once you get down into getting rid of all of the refined foods, so that they can stop thinking about it in terms of throwing terms like fad diet around and start thinking about it in terms of a mechanism, and maybe we'd actually get them interested that way. And of course, it's difficult to convey, even when you've got good science. It doesn't always get through to the front line. I can't tell you who said this and to who, but they said, that's easy for you to say. You have access to all these papers, all these studies. The rest of us go by guidelines because we don't. I mean, where do you get all these studies anyway? So it's a communication issue as well. So, quick review of the hypothesis. We know that the vagus nerve plays a really important role in signaling about diet to the hypothalamus in the brain, and we know that it gets inflamed and that alters its ability to do accurate energy homeostasis early in obesity models. We know that those obesity models are pretty much dependent on the presence of the GI microbiota, and we also know that the innate immune response to that microbiota is now implicated in all those conditions. I ripped this from a Nature paper recently, so you can see it's been taken seriously. We know the oral microbiota is profoundly altered by the presence of flour and sugar, and that it's the innate inflammatory and immune response to that altered microbiota that produces periodontitis. So it seems sensible to suggest that one swallow away from that inflammatory effect in the mouth, we may have a similar inflammatory innate immune response to a similarly perturbed microbiota in the upper gut, and that through proximity to the crucial functioning of the vagus nerve and to the microbiota, the rest of the immune system itself, and the importance of the microbiota in the immune system, that may be the main generator of an awful lot of Western disease. So the hypothesis is consistent with the virtual absence of Western disease in hunter-gatherers, the way that low carbing seems to work, the way that ancestral diets seem to work, but in a macronutrient independent way, the relative resistance of Europeans to metabolic syndrome through 10,000 years of downstream adaptations to the low-level gut inflammation from flour, the association of dental and systemic health, the way that real food diets seem to work best, but when you supplement with their pieces for fiber and supplements, you don't seem to reproduce the effects of an ancestral diet. You really need to stop dropping flour and sugar into the bioreactor next to your immune system. And the association of autoimmune and autoinflammatory conditions with Western diets. I'd like to say a big thank you to the Ancestral Health Society for having me. I'd like to thank the Ancestral I'd like to thank the Ancestral Health Society of New Zealand because they've been very supportive over the years and they're financially supporting me here. So that's how I'm able to be here. And I'd like to thank my longtime friend and colleague, Dr. Andrew Samis, for years of great conversations. And he recently did a sterling job up in Canada with the Canadian Heart and Stroke Foundation. He helped overhaul their dietary guidelines on dietary fat towards a more whole food message, so he's doing a great job up there. Thank you very much.
Thank you for that great talk. We have plenty of time for questions, so again, please come to the center microphone. I'm going to actually steal this opportunity to ask the first question, and I was curious to hear your opinion on refining whole foods, particularly blending. So if you take whole foods and you put them into a blender, in your opinion, does that refine them too much?
This is a complete cop-out, but there's no way to tell. You'd have to suck it and see. Against a background of a diet that you know is working for you, give it a try, see what happens. It's too complex to even venture a guess.
Hi, great talk. What have you seen with fecal microbial transplants? David Perlmutter especially, I've noticed, has really been impressed with what they can do. Have you seen any results, and might it be the fastest way to heal certain conditions?
Certain conditions, absolutely. Things like C. difficile, of course, it has a great track record, and they were working on that at GI Diseases at Queen's when I was working there with their repopulate. If it's anything that's come out in the literature in the last year, I may have missed it because they kind of keep us busy at med school, so I haven't been keeping up to date with the literature as much as I would have done. It wouldn't surprise me at all that it's a great, by dropping the whole ecosystem back in, you can achieve so much more than you can just by dropping in a few lower concentrations of probiotics that may get probiotics that might get outcompeted.
Mm-hmm.
So.
And if you were to consider doing it yourself and getting past the repulsion, I guess, of taking that capsule, where would you think would be the single best person in the world to find that donation?
Oh, that's a good one. I don't know. Of course, you've got to check out a lot for safety reasons as well before you start doing things with other people's poop. I don't think it's usually a capsule. It's usually up the other end.
I really liked your point about draining the swamp, because I think that it's not very plausible to me that our guts are optimized for some kind of element that wasn't continuously available for a really long time. And just cutting back on all the noise, I think, allows our healthier microbiome to grow. Can you speak a bit more about that?
It's difficult, really, because I get hives if I go too far into speculation, which I know I'm very guilty of anyway. One thing that's crossed my mind is whether, with things sitting in a biofilm, so they're more resistant to changes outside, that they're sharing genetic information, is it possible that just by making them go completely without food for a while, they have to drop some of their defenses? But it's your guesswork. I'm not a microbiologist, so you'd get much more sense on this out of Grace or someone.
It ties in with some of the positive things we see in the not-abiotic animals as well. Thank you.
Oh, no worries.
Thank you for that talk. That was excellent. As one of the few dentists around here, I get very excited about when you guys mention the mouth.
I like dentists.
Awesome. I have two questions. My first one is sort of clarification. It's my understanding that most of the gut, the oral flora don't really survive an adequate stomach acid, so we're not talking about the oral flora seeding.
Well, it will be to an extent, because it's just a question of with every swallow, even if you've got 99.999% mortality, sooner or later some of them are going to get through and seed. The data that I've seen, I can't remember the stuff off the top of my head from the duodenal study, because that's a lot more complex, but it's usually just cited as Lactobacillus streptococcus. and you have a lot more listed up in the oral. So there's definitely a pinch point at the stomach, but some of it's getting through.
Clearly, I mean, with the Western diet, we have a tendency to have lower stomach acid anyway, so all bets are off with, with SIBO and things like that. My next question is, have you come across anything in the literature that might look at the oral, swabbing the oral microbiota, culturing it, and using that as a proxy for something like SIBO or some dysbiosis?
I'm not sure about people may have been looking for SIBO, but that research probably passed me by because there's so much more of it. There was definitely something on looking at the oral microbiota in obesity. I don't remember the specifics, but there was one particular either species or phyla of bacteria that if it was present, you could predict that person would be obese with about 98% certainty. Again, whether that's causal or it's just another bacteria that likes those conditions, it's hard to say.
Thank you very much.
Thank you.
Thanks. I very much enjoyed your presentation.
Thank you.
My question is about the gut-brain connection. And so if the vagal nerve is connecting only to the small intestine, what is the connection then to the large? Is it the enteric nervous system?
Oh, blimey, now I'm back in my anatomy classes. I think there's a similar parasympathetic pelvic nerve, I think, that goes, but it's not something that's been looked at intently with the point of view of obesity, for perhaps obvious reasons. I'm pretty sure, the brain knows what's going on in the colon as well. It's just not been documented as well from the point of view of obesity.
Okay. Thanks.
So my question has to do more with performance perspective, which is going to be obviously a little different than a health perspective, but there seems to be benefit to simple sugar post-training for people who are trying to perform at a high level. But there also seems to be potential issues with gut damage, all that type of stuff from training with a lot of intensity and volume. Do you have any thoughts in terms of potentially mitigating the effects of simple sugars post-workout for someone who may have gut issues but wants to intake, a bunch of dextrose or Vitargo or something post-workout?
I don't know. I've not considered that one before. I mean, work out very, very hard to try and get things going as anti-inflammatorily as you can. Do it while you're young. I don't know. Maybe no one here is going to like this, but drop the fat into the floor, like in the studies in the 70s that Kempner was doing. If you drop the fat in the floor, maybe not as much of it would get across.
Sure. Yeah. I mean,
but I mean, how that in turn would knock on to performance training, I don't know.
Yeah. I mean, from sort of anecdotal perspective, a lot of people who do very well with just like high volume, high intensity training basically live on a glucose strip. They're just like protein powder and simple sugar and no fat all day.
Would mashed potatoes work?
might be tough to take those down after something.
Yeah, no, I understand. Yeah, the logistic problems that people have got. I'm too sedentary for this to be a direct problem for me.
Cool. Thank you.